Good morning, everyone. Today we have Dr. R.S. Deepak here. He's the Associate Professor, Department of Psychiatry, MR Medical College Kalaburagi. He has done his MBBS from JJMC Davangere and DPM from Forebrain, MD from Kasturi Medical College Kuntur. He has also done MBA Hospital Administration from ICFAI University. Currently, he is working as Associate Professor at Basaveshwara Medical College, Chitradurga. He has 8 papers published in peer-reviewed national and international journals. His area of interest is community psychiatry, and so far he has conducted more than 250 radio programs to create mental health awareness. Also done more than 100 public talks on mental health in various forms. Over to you, sir. As Sir added, he also plays very good cricket.
Thanks for the warm introduction, madam. At the outset, I'd like to thank the organizing team for this opportunity. It's a privilege to come to Gulbarga. As you all know, my aunt stays here. Okay, let's start the topic. It's Neurobiology of Sleep. First, when they gave this topic, I was thinking I should not make everybody sleepy. It's a little complicated. We'll try to decode it as simplify it as simple as possible for the general audience. Thank you. Okay, what exactly is sleep? Sleep is a resting state of our body, which is characterized by reduced response to external stimuli, lack of mobility or a slight mobility, characteristic sleeping posture. Everybody has a different posture on their own. Nobody, and in fact, it keeps changing at times. Reversibly unconscious state. It's a very important point, probably. If sleep had to be a lot more, if it is not irreversible, it is death, isn't it? Most of us humans sleep with eyes closed, unlike some animals. So all these characteristics should be there for us to have a very good sleep. It seems very simple that we all sleep just like we are fatigued, we sleep. But it involves a lot of different neurotransmitters. A lot of brain centers have to work for us to have a good sleep. So now the whole arousal spectrum, what we conceptualize as of today is to maintain a right proportion of wakefulness, then only we can function. We can probably do day-to-day activities. We can concentrate on our external sensory stimulation. Our cognitions will work only when there is a right proportion of wakefulness. At the left side of the spectrum, if there is, if we are not aroused properly or we are a little sleepy, the cognitive dysfunction starts happening. Probably we may not be as attentive, or we may not learn, or we may not be contributing adequately. Similarly, if there is a little hyperarousal, that also leads to cognitive impairment. The whole spectrum from daytime sleepiness to excessive hyperarousal would lead to probably over analyzing our surroundings, which might end up having fearful symptoms, or to an extreme spectrum, we might have some psychotic symptoms also. So this is the whole spectrum, how we are conceptualizing wakefulness and sleep spectrum. Coming to wake circuit. See, for us to be awake, the most important neurotransmitter is orexin, which is released from hypothalamus. It in turn stimulates monoamine centers like locus coeruleus, releasing norepinephrine, raphe nucleus, releasing serotonin, and basal forebrain releasing acetylcholine, nucleus accumbens releasing dopamine. So there is a huge interplay of wakeful neurotransmitters which will be functioning for us to keep us active, awake, attentive, and function our day-to-day activities. I'll be explaining each of these centers in detail. Coming to acetylcholine. Acetylcholine is predominantly released from basal forebrain and brainstem. It has two different pathways. Basal forebrain cholinergic neurons is active in wakefulness and REM sleep. During NREM stage, acetylcholine is not triggered. Leads to fast EEG rhythm due to projections in cortex and hippocampus. Reduces activity due to inhibitory cortical interneurons. Feedback mechanism. So the other pathway is from pons, where projections to subcortical regions, thalamus and lateral thalamus and forebrain, promotes cortical activation by releasing acetylcholine. So one is from the pons, it will activate awakening. The other pathway would stimulate sleep. Histamine is again an important neurotransmitter for sleep. I was talking about orexin. Orexin would not work directly. Orexin in turn triggers histamine. Histamine is released from tubulomemory nucleus, projects to whole of forebrain and brainstem. It in turn stimulates all the monoamines. Coming to orexin. See, orexin is an important neurotransmitter which would stabilize sleep. As such, orexin is not a stimulating neurotransmitter, but it is important to stabilize our wakeful state. What exactly orexin does? See, orexin would directly stimulate ventral tegmental area, releasing dopamine. Again, orexin would also stimulate locus coeruleus, releasing norepinephrine and serotonin. All these, along with acetylcholine, would maintain wakeful state. Similarly, orexin has a role in appetite also. So, orexin is inhibited by VLPO, which is a GABAergic center. So, all these things, if orexin is not functioning well or somebody has a hypothalamus injury, probably the sleep can't be stabilized. If orexin is not there, newer drugs, whatever they're talking is orexin antagonists. So I was talking about serotonin, norepinephrine, dopamine, acetylcholine, orexin, histamine. So there are a lot of neurotransmitters are required for us to be awake, whereas only one neurotransmitter is enough to make us asleep. That is GABA. So GABA is released from VLPO, which in turn will inhibit all these wake promoting neurotransmitters. So by nature or biology as it stands, our brain or the nature would want us to be awake more rather than asleep. So there are huge play of neurotransmitters which have to be active for us to be awake, and our brain tries maximum to keep us stay awake. Again, what is the role of thalamus? Thalamus has two different neurotransmitters. It all has glutaminergic thalamocortical projections also, which would relay sensory, motor, and limbic information to cortex. And it has a reciprocal GABAergic neurons also, which would make us cut down all the sensory stimulation and make us asleep. So to simplify it, see what exactly happens. See, when we are awake, thalamus is a highest center for all the sensory filtering. Whatever, see, we, you, me, everybody is awake here. We're getting multiple sensory stimuli. You are trying to hear to me. Whatever auditory stimulus you're getting, you're sitting, probably you're not sitting on a comfortable chair. There is a little bit of pain or there are some noise in the background. All these things are there in the surroundings. When we are awake, most of it, because of the glutaminergic tone in the thalamus, most of it is not filtered. Everything goes to cortex and it tries to process information. And again, we will try to take what is important at this point of time will process it and which would make us awake. Okay. Exactly what happens is opposite in the sleep. Thalamus, even when we are asleep, there might be some noise, there might be so many other sensory stimuli, but most of it is filtered off because of the GABAergic inhibition. This GABAergic inhibition is very much essential for us to stay asleep. If at all, say, just presume if the GABAergic inhibition is too much, nobody could wake us up properly. We would end up comatose. Or say the GABAergic inhibition is very low, even a minimal stimuli, probably a pin drop, will also make us awake. So we all should salute GABA for us, GABA to make our nights very happy. So to summarize, see, as I was already talking in the previous slides, acetylcholine is predominantly active in wakeful state or in REM sleep. Monoamines, again, are very much active only in wakeful state. Monoamines in the sense dopamine, norepinephrine, and serotonin. Orexin, as I was already telling, is a main neurotransmitter which stabilizes sleep. It is probably the kingmaker of our wakefulness. Coming to VLP or GABA, GABA is very silent when we are awake. GABA is active only during our sleep. Okay, so all the nuts and bolts are over. Now, how does sleep exactly happen? There are two different processes for us to sleep. One is sleep homeostasis, which is also called as process S. The other one is biological clock, process C, circadian rhythm, regulate timing of sleep and wakefulness. These two pathways work in tandem for us to go to sleep. What exactly is circadian rhythm? See, whenever there is light in the surroundings, and I'm not even talking about sunlight, even our LED lights, the light which we are very fond of these days is screen light, either coming out of our telephones, mobile phones, or LED screens. So whenever the light falls on the eye, retinoganglion cells are activated, which will be relayed through retinohypothalamic tract. It stimulates suprachiasmatic nucleus, which in turn inhibits the release of melatonin from pineal gland. When the light fades, say probably around sunset or sundowning time, the light has gone away. Thereby, the suprachiasmatic nucleus is inhibited, which would in turn facilitate release of melatonin, which would stabilize sleep and make us sleepy. Generally, melatonin secretion starts somewhere around 8 o'clock in the night and it peaks around midnight. Okay, coming to wake circuit. So what I was already telling, we are trying to summarize here. Suprachiasmatic nucleus during the light phase would stimulate release of orexin. Orexin in turn releases histamine. Histamine spreads all across the forebrain, hypothalamus, midbrain, releasing acetylcholine for basal forebrain, dopamine from ventral tegmentum area, norepinephrine and serotonin from locus coeruleus and raphe nucleus. These are wakeful neurotransmitters which will keep us awake. Exactly opposite happens as the light fades, norepinephrine and serotonin will be built up, which in turn, by feedback inhibition, it would stop the secretion of orexin. Once the orexin is stopped, the VLPO will get activated. A lot of GABA is pumped into the system, which would make us asleep. Coming to other process S, sleep homeostasis. This process is very simple. The more number of hours you are awake, more fatigued you are, it is the need for sleep, more or less like a hunger drive. It is a sleep drive. As we are number of hours we are awake, adenosine is secreted. Accumulation of adenosine would disinhibit the VLPO, releasing GABA. It would make us asleep. Clear? There are two different processes, circadian rhythm and processes, which would work in tandem to make us sleepy. Okay. How does this flip happen from wakeful state to sleep state? There are three different things which would work here in a flip-flop switch. How do we switch off to sleep and how do we get up from sleep? Importantly, orexin, VLPO, tubulomammillary nucleus, which would secrete histamine, locus ceruleus, and raphe nucleus. These three work in three different mechanisms for us to flip. What exactly happens when we are in awake state? Orexin, with the help of would stimulate tubulomammillary nucleus releasing histamine, which would in turn stimulate locus ceruleus and raphe nucleus releasing norepinephrine and serotonin. These neurotransmitters spread across the cortex and inhibit VLPO, thereby we stay awake. So it's a triple whammy. These three should work in tandem for us to keep us awake, or this switch will be on when we are in awake. So when we are asleep, what happens is VLPO will become dominant, as I was already telling, because of the GABA input. VLPO will directly inhibit these monoamines. VLPO will also inhibit orexin. What you could make out from this slide is orexin does not have a direct inhibition of VLPO. Orexin would not inhibit GABA. But whereas GABA, once active, it can inhibit everything in the sleep circuit, in the awake circuit. This is how our flip from sleep to wake and wake to sleep happens. Coming to the biology would not be complete without me telling EEG findings. What are different stages of sleep? One is wakeful stage, which we call as stage W. There is an NREM stage called as non-rapid eye movement sleep. Recently, the categorization, they have changed it to N1, N2, and N3. Previously, it used to be NREM1, NREM2, NREM3, NREM4 also. But now they have categorized as 3 stages. And rapid eye movement sleep, REM stage. So generally, for us, around say adults, this sleep cycle would take 90 minutes to 120 minutes. And they probably for children, the sleep cycle would take a longer period. Thereby, they end up sleeping more time. For us, anybody for that matter, at least 4 complete sleep cycles are required for us to feel active or to have a satisfied sleep. Whereas in children, the same sleep cycle would take 3 hours to complete. They end up sleeping more than 12 hours. As we age, the NREM 3 stage 3 would reduce and their sleep cycles get shortened. So probably say somebody around 70, 80, they would sleep for 5 to 6 hours. They also complete sleep at least 4 sleep cycles, but their sleep cycles would happen faster. Coming to stage 1, wakefulness, where when our eyes are open, we will have low voltage alpha waves. Eye movements and blinking present. Tone of muscles are quite tonic. In one stage, the EEG will have theta activity, 4 to 7 hertz. Eye movements are slower. Tone would start reducing. So 5% of our bedtime we'll spend in stage 1. Coming to stage 2, 50% of our time we are spending in this stage, important stage. The sleep is becoming a little more, getting into depth. So muscle activity reduces further. The sleep characteristic sleep EEG changes would happen in N2. Sleep spindles will happen, K complexes will happen. The sleep spindles and K complexes are result of, I was talking about antagonizing mechanism. Glutamate is there and GABA is there. Because of this antagonistic action, these sleep spindles and K complexes will happen. Stage 3, N3, 25% of our time is spent here. REM sleep. Again, it is a little different, probably a lot more closer to wakeful state, but we are at the depth of our sleep. The tone, all our metabolic parameters goes up to a little more extent, unlike NREM sleep. So, all the EEG recordings are predominantly because of excitatory and inhibitory changes in pyramidal neurons. This I've already told. During wakefulness and REM sleep, the potentials are desynchronized, resulting in low amplitude, fast activity. Whereas in NREM sleep, the signals are synchronized, resulting in high amplitude, slow activity. To summarize, there are interplay of lot of excitatory neurotransmitters play. Example, histamine, orexin, norepinephrine, dopamine, serotonin. Whereas for us to make a sleep, GABA plays a huge role. And again, see, I was trying to tell circadian rhythm. Any light is good enough to shut down our melatonin secretion. Off late, we end up getting a lot of patients with sleep problems because of one idiot box called as mobile phone. It emits a lot of blue light, which directly inhibits secretion of melatonin and would make us. It would put an image to our brain as if it is still very sunlight is active. So our circadian rhythm goes haywire. We would not end up sleeping well. If we are not getting good sleep, we are probably inviting a lot of other medical comorbidities and psychiatric comorbidities. So I hope I made sense. Thanks for the patient hearing. Thank you.
Thank you, sir. Thank you for enlightening.
I know biology. I can understand it right now. I have not concept. I came to know this is the subject I am looking at. As we know, the preoptic cells in the preoptic area and lateral thalamus group, and as you know, the neurotransmitters working there, and also metabolic region. These sleep, you will say, also have active neurocell activity. It is not a passive or strong. It is an active one. And forms and organs are activated when there is the REM sleep and all. And what is that is what I understand. And lot of development has come, you are unable to work. So much of them, what I understand. This is the thing. Where active stage of the neural cell activity. So this is what I want to say.
Thanks, sir.
Thank you, sir. It was a very nice and brief presentation, and at the same time, in depth about the whole subject. I hope it was very useful to the postgraduates here. And any questions, anyone? Any postgraduate student want to ask any question? There are no questions. Okay. But I think the subject was so interesting and in depth that nobody was asleep during the presentation. So it was a nice presentation. So thank you. Yeah. Yes.
Deepak, thank you for a very enlightening update on neurobiology of sleep. two things. One is all new psychopharmacology is targeted at orexin and antagonistic drugs, which are likely to come in the next 5 years.
Yes, sir.
In India also. That's one. And second thing is all behavioral addictions, social media addictions have something to do with the neurobiology part of it. So people should understand how much mobile they should use and what that they should see during night if they want to have a good sleep. Those are the two, I think personally, those are the two take-home points from your side.
Thank you, sir.
Thank you.
Sir, I have a question for you. In your practice, sir, all these sleep supplements, which I would call the melatonin or other things, how useful they are for your patients because compared to the other benzodiazepines. So, do you recommend giving the supplements or prefer benzodiazepines?
See, basically, when you're talking about insomnia, probably there are next speakers who would highlight on a lot of things. See, most often as we psychiatrists, we tend to see sleep as probably a pharmacological intervention is required. Most of the times, just talk to them and take their routines. As I was telling, this behavioral addiction, and as Sir was rightly pointing out, everybody who has a smartphone is addicted to smartphone. So, and most of us would not get a lot of time in the day. We end up spending a lot of time at night seeing some or other nonsense. All of us are part of huge network of social media platforms where we end up spending a lot of time unnecessarily. So, getting to the details of why they are not getting sleep or other reasons for their sleep, their routines, how well we could probably set it right by non-pharmacological means would be my primary goal. Say after all that still there are we are not able to achieve good sleep hygiene, or even despite maintaining good sleep hygiene, if they're not sleeping well, benzodiazepines can be used. There are, like, say, H1 antagonist drugs can be used. Small amount of antipsychotics, second generation antipsychotics in lower doses can be used. Tricyclic antidepressants can be used. I feel probably this will be highlighted in the next this thing. But again, I don't want to probably pour water on everything. We have not found an ideal hypnotic as yet. A lot of issues with our sleep medication. Probably they would work for some time, then the same drug may not work, or the patient end up pill popping, or probably tolerance would they develop. There's a lot of issues, and a lot of research is going in sleep and neurobiology of sleep, or pharmacology of sleep is for the same reason, because we have not reached a stage where we have at least nearly an ideal drug for the sleep.
Thank you so much. Any more questions? Thank you, sir. Thank you for highlighting that sleep is not just a state of passive state, but a much more active process than we think. Now I request the chairpersons to present the momentum to the speaker.